Homer Smith was a visionary. He created the field of renal physiology.
He inspired generations of physicians.
The Homer Smith Award of the American Society of Nephrology was the first award given, and it reflects Homer Smith's dominant role in understanding the physiology of the kidney.
Smith led the Department of Physiology for decades at NYU, and it was Smith, probably more than any other person, who defined the modern concepts of glomerular filtration, tubular excretion, and also secretion.
He mentored an entire generation of people who followed him and created the principles on which the field was built.
My father was always dressed in a suit. People always saluted him wherever he went. And I understood he had an important position at NYU. He was always well admired and very well respected.
One of his great contributions is that he brought to NYU techniques and information that he had gathered by studying fish. Many of those experiments were carried out in Mount Desert Island.
The Mount Desert Island Biological Lab played an extraordinary central role.
He had been the chairman and the head of that lab for many, many years.
Smith realized that marine organisms gave him powerful tools to study the evolution of the kidney.
He was into philosophy, music, science in the broadest sense, religion.
To read him, it sort of opens your mind to other areas and it fires the imagination.
I think that every nephrologist owes a great debt of gratitude to the scientific contributions of Homer Smith.
When he said something, it turned out to be correct because he was very precise in what he did. And that's a lesson for all of us.
The American Society of Nephrology presents the 2022 Homer W. Smith Award to Dr. Jeff Sands in recognition of his major contributions to the understanding of the mechanism of urine concentration. Dr. Sands is director of the Division of Renal Medicine and Juha P. Koko Professor of Medicine at Emory University School of Medicine in Atlanta. He has also served Emory as Executive Vice Chair of Medicine and Associate Dean for Clinical and Translational Research. A graduate of Boston University School of Medicine, Dr. Sands completed his residency at the University of Chicago, followed by a research fellowship at the National Heart, Lung, and Blood Institute. He then completed a clinical nephrology fellowship at Emory and joined its faculty in 1989. Dr. Sands' research focuses on the molecular physiology of urea transporters, aquaporins, and the urine concentrating mechanism. His team has worked to translate these basic research findings into novel therapies for nephrogenic diabetes insipidus. Their research showed that vasopressin, a key hormonal regulator, not only affects water transport within minutes, but also stimulates urea transport. The group also investigated whether there are non-vasopressin-mediated pathways that increase urea and water transport as a potential strategy to treat congenital NDI. This work led to the discovery of an investigational drug that increases urine concentrating ability and the formation of a startup company to advance work on the drug. Dr. Sands has authored 175 manuscripts and over 100 invited reviews or book chapters, and co-edited a book. He has given numerous invited lectures at scientific meetings and universities both nationally and internationally. He has received funding for his research from the National Institute of Diabetes and Digestive and Kidney Diseases, as well as the American Heart Association. Dr. Sands has also been active in training the next generation of renal researchers, both postdoctoral fellows and undergraduate students. Among many examples of his professional service, Dr. Sands chaired an ASN annual meeting program committee and the American Heart Association Kidney Council, and was a member of the NIDDK Board of Scientific Counselors. A past president of the American Physiological Society, he was editor-in-chief of the American Journal of Physiology, Renal Physiology. Dr. Sands has received several honors for his work, including the Distinguished Alumnus Award from Boston University, the Barry M. Brenner Endowed Lectureship from ASN, and an honorary degree from Aarhus University in Denmark. Jeff and his wife Abby reside in Atlanta, Georgia. They enjoy traveling the world together and spending time with their children Jared and Jenna. Dr. Jeff Sands has earned the respect of colleagues and students worldwide for his outstanding contributions to the science of nephrology.
Congratulations to Dr. Sands. I would like him to join us on the podium. He will also present on urea transport to nephrogenic diabetes insipidus using physiology to develop novel therapy. Dr. Sands. Yeah, I'll get it.
Well, good morning, everyone. Let me start by offering my sincere thanks to the American Society of Nephrology and the Awards Committee for bestowing the Homer W. Smith Award on me. It's a tremendous honor and very humbling for me to receive this incredible award, so thank you. As you saw in the bio, I guess you now know what the title of my talk is going to take us through. Oops. Okay, what did I do wrong here? There it goes. Doesn't seem to want to advance. There we go. Okay, I do have some disclosures, particularly that I am one of the founders of a small startup company, and we've had some support from both Arinia and Otsuka to our laboratory at Emory, not for me personally. Some learning objectives, which are basically also the outline of the talk. We will review urea transport and its role in urine concentrating mechanism. We'll review non-vasopressin signaling pathways that regulate urea and water transport, and we'll review nephrogenic diabetes insipidus. So I have many people to thank. Of course, I started by thanking the American Society of Nephrology, but I'm really delighted that my family is here with me today in the audience. My wife Abby, my son Jared, and my daughter-in-law Alex, and my daughter Jenna, and her fiance Saul, and they're all sitting in the front. I want to thank them for all their support and love over the years, especially all those times where they probably thought I was spending too much time working and not enough with family, which I probably was. I want to thank my parents, Jean and Joe Sands. They are not here, but they are watching this in Canton, Massachusetts over the live stream. I got my start in research as an undergraduate in the laboratory of Dr. Ryan Bucas at Harvard Medical School. And it's why to this day I'm a big supporter of programs that support undergraduate research, such as the ASN Kidney Stars program and the KUHR 25 programs. I owe a lot of gratitude to Dr. Bucas, who took me in as an undergraduate. You can see this eventually led to the first paper that's shown there. I was already involved in urea way back then. And I came to my first ASN meeting way back in 1977. So Sue, I got here even before you did. Back then it was just called the ASN meeting. There was no renal week, kidney week. It was just the ASN meeting. But that meeting was particularly important, probably more than presenting the poster, because Dr. Maurice Berg won the Homer Smith Award in 1977. He was the pioneer of the isolated perfused tubule technique. You can see a picture of Dr. Berg sitting in front of a dissecting microscope. He was also ASN president from 80 to 81. Shown on the right-hand side is a perfusion apparatus, very similar to what I used when I subsequently went to do my fellowship at the Lab of Kidney and Electrolyte Metabolism. And underneath is a schematic of a perfused tubule where you can see, not sure where the mouse went, but anyway, you can see the tubule where it's microdissected and mounted on pipettes, and you can put fluid into the lumen of the tubule, collect it out the other end, and make a precise measurement of kidney transport. And it remains the gold standard to this day. And sadly, Dr. Berg passed away in April of this year. And in tribute to Dr. Berg, during the rest of my talk, I'm going to focus heavily on perfused tubule data that we have generated rather than other techniques that we've employed. Now, I was so inspired by Dr. Berg's Homer Smith Award lecture that I applied to his laboratory and was fortunate to be accepted and became a postdoctoral fellow under the mentorship of Dr. Mark Knepper. Mark is here today. Mark also won the Homer Smith Award in 2002. And as a fellow, we published the paper that's shown here on this slide. This was actually the first paper in which anyone proposed having a urea transport protein in the kidney. Now, whether we were the first to think about it, I don't know, but we were the first that the reviewers allowed it to go through and get into print. So I guess we get credit for it in that regard. I also owe a great deal of gratitude to three individuals at Emory who served as mentors. Dr. Coco is sitting here in the front row. He was the chair of medicine who brought me to Emory. Dr. Bill Mitch, who's not here today. They are both past presidents of the American Society of Nephrology, and Dr. Douglas Eaton, who I believe is here, and he is a past president of the American Physiologic Society. So these three individuals offered me guidance, career guidance, scientific guidance, and also, as you can see, they're all past presidents of societies. They emphasize the importance of national service in addition to doing science. So, and then Dr. Janet Klein, who's in the audience, we've worked together for over 25 years. Virtually everything I'll show you today, Janet had a hand in, or two hands, depending on the nature of the experiment. You can see our first joint paper there. It's actually on aquaporin 2 and not on urea, and we're standing in front of a large jug of water. Finally, and this is completely unreadable, but these are all the people I've had the privilege to co-author a paper with over the years. I hope I got it right, but since nobody can read it, I guess it won't matter if I messed it up a little bit. But it takes, science is a team sport, and I've had great teammates over the years. So let's turn now to the science. And shown here, and this picture was actually in the video, our charge distribution of both urea on the left and water on the right. And everybody knows that water is a polar molecule, H2O. Well, when you look at the electrostatic charge distribution of urea, C double bond O in the middle, two NH2 groups to the side, it looks just like water. It's a polar molecule. I want to thank David Weiner, who gave me these pictures. And so urea, like water, has a low permeability across artificial lipid bilayers. But urea, it's not zero. So it does equilibrate across membranes, but it does so slowly. But in the kidney, the transport is simply too rapid. It can't happen by simple lipid phase diffusion, hence the need for an actual urea transport protein in the intermedullary collecting duct in particular, as well as in the vasa recta. Urea has been known to play a key role in the urine concentrating mechanism for almost a century. The studies by Gamble and colleagues described an economy of water and renal function referable to urea. This paper, based purely on clearance measurements, Robert Fenton and Mark Knepper did studies in knockout mice in 2006 that really confirmed the findings of Gamble and extended them from his early publication. Protein deprivation reduces maximal urine concentrating ability in every mammal that's been studied. Now, these are not the low protein diets that we prescribe for patients, but this is true protein malnutrition. And many urea transporter knockout mice have been made. You can see the different versions here. Regardless of which one is knocked out, all of the mice have a decrease in concentrating ability. The phenotypes varies depending on which one is knocked out, but the bottom line is if you lack urea or urea transporter, you cannot concentrate maximally. Now, this is a schematic of the urine concentrating mechanism. This is my view. You've got your loop of Henle and your collecting duct. Apologies to all the people who love the glomerulus, but it really isn't the central player in urine concentration. The single effect for concentration comes here from the sodium-potassium 2-chloride cotransporter in the thick ascending limb, which combined with the potassium coming back leads to the secondary active transport of sodium chloride out of the lumen of the thick ascending limb and into the medullary interstitium, where by countercurrent multiplication, the osmotic gradient increases with depth. In the presence of vasopressin, it activates aquaporin 2 water channel, increases apical membrane accumulation, and you can have reabsorption of water down its osmotic gradient. The urea transporters come in here, in the terminal intermedullary collecting duct where UTA1 and UTA3 are present, and urea can be reabsorbed down its concentration gradient to add to the urea content and the osmolality in the deep medullary interstitium. This is the paper that I showed earlier with Mark Knepper, in which we showed that vasopressin increases urea permeability in the terminal IMCD. You can see here in the initial IMCD where there are no urea transporters, you have a low basal permeability, no effect of vasopressin. In the terminal IMCD, there's a high basal permeability, and it goes up over 4-fold with vasopressin. In subsequent studies, that I don't really have time to show you the data on, we went on to show that vasopressin increases UTA1 phosphorylation at 2 serines, 486 and 499, and we also showed that vasopressin increases UTA1 apical plasma membrane accumulation. Now, how does vasopressin signal? Well, it increases urea and water permeabilities through the cyclic AMP-PKA pathway. So vasopressin, this red square, binds to the V2 receptor in the basolateral membrane of the intermedullary collecting duct. It activates adenylyl cyclase, generates cyclic AMP, activates protein kinase A, which ultimately leads to the phosphorylation and apical membrane accumulation of both UTA1 and aquaporin 2. But are there other non-vasopressin pathways that can increase urea permeability? And this is a prelude into where we ended up near the end. If you're going to treat nephrogenic diabetes insipidus, we needed to find something that basically bypassed the traditional vasopressin PKA axis. So we had a clue. This is actually one of the first papers I published when I was at Emory with Dirk Schrader. And we found that hyperosmolality increases urea permeability independently of vasopressin. And that's shown here. We simply added sodium chloride to the perfusate and bath of perfused tubules, and you can see that as you raise osmolality, urea permeability increases at least up to 690, not further at 890. In the dotted lines, we actually started here and worked backwards to make sure there was no osmotic damage to the tubule. It is synergistic with vasopressin as well, and subsequently we showed that osmolality also phosphorylates UTA1, but at a different serine, at serine 494, and increases apical plasma membrane accumulation. So which signaling pathway mediates the effect of hyperosmolality? Well, we looked to see did it work through cyclic AMP, and it does not. There was no effect on cyclic AMP production. And then in collaboration with Robert Starr, we did this study in which Robbie perfused a tubule and increased osmolality to 490, and you can see a nice increase in intracellular calcium in this perfused tubule. Well, intracellular calcium suggested perhaps this is protein kinase C. So we used PKC alpha knockout mice that we got from Jeff Mulkenton in Cincinnati to pursue this. And these mice have a urine concentrating defect. You can see the normal mice are over 2,000 and the knockouts under 1,000. Janet Klein went on to look at UTA1 protein abundance by Western blot in the inner medulla. And you can see a representative here. It runs as 117 and 97 kilodalton glycoproteins, wild types, much less in PKC alpha, about a 50% reduction, so it was consistent with the decrease in concentrating ability. To take it one step further, Anna Wang perfused tubules from the mice and looked at whether hyperosmolality had an effect. She first did wild-type mice, and just like in normal rats, simply increasing osmolality increased the urea permeability of wild-type mice. But when she did PKC alpha knockout mice, no effect whatsoever of increasing osmolality. Now, perfusing rat IMCDs is hard, perfusing mouse IMCDs is harder, and perfusing knockout mice IMCDs is even harder than that. So this is a tour de force by Anna to do this study. So these data, looking at hyperosmolality and PKC alpha, did show that there is a non-pKa pathway that increases urea transport. But hyperosmolality is stimulated through NKCC2 in the thick ascending limb under the control of vasopressin. So we still have a problem. Hyperosmolality alone cannot be the answer for treating nephrogenic diabetes insipidus, because how'd you get hyperosmolality if you don't have vasopressin? So we needed something else. And we thought about another kinase that perhaps could be the key player. So before we get to that, we'll review a little bit about nephrogenic diabetes insipidus. These patients produce very large quantities of dilute urine due to kidney insensitivity to vasopressin. As shown here, here's the tubule lumen, a collecting duct cell, the interstitium, and you get nephrogenic DI either because in fact, in adult medicine, most cases are acquired primarily due to chronic lithium use. Although not shown here, lithium interferes with cyclic AMP production. But the congenital forms, and there are two, are very rare. It's a pediatric orphan disease, actually an ultra-rare disease, and about 10% of cases are hereditary. 90% from mutations in the V2 receptor, shown here, 10% from mutations in the aquaporin-2 water channel. The V2R mutations follow an X-linked pattern of inheritance, so it is only the boys who are affected, whereas the aquaporin-2 mutations are mostly autosomal recessive, although some are autosomal dominant. So kids with congenital NDI can produce up to 20 liters of urine a day, and they must drink an equal volume of water to avoid dehydration. If they do not do that, they can get cognitive impairment from severe episodes of dehydration. Just to put this in perspective, here's a boy drinking water. Maybe a normal, healthy boy would be told to drink 1 liter of water a day. We know we can't get our kidney stone patients to drink 2 liters a day, despite the pain that kidney stones cause. This is what this poor kid with NDI has to drink. 20 liters of water, and he's constantly needing to void. So to look at how we might come up with a therapy for NDI, we wanted to ask this question, which was, is it simply something that we need to fix aquaporin-2 getting to the membrane, or do you need UTA1, urea transport, as well? And so we used UTA1, UTA3 knockout mice. These were a gift from Mark Nepper. And T.T. Allori did this study where she took first wild-type mice, Here's their osmolality, 3,000. You give them vasopressin for a week, it goes up to 4,500. The UTA1A3 knockout mice make a urine of around 800, which is basically what just an outer medulla can do without that intermedullary contribution where urea plays a role. And if you give the knockouts vasopressin for a week, no benefit. They have nephrogenic diabetes insipidus. What about aquaporin 2? Turns out it's perfectly normal. Here's a Western blot of aquaporin 2, a 29 kilodalton band, and a smear from 35 to 45. You can see a nice increase with vasopressin. This has been shown by many laboratories many times. The knockouts have a little less UTA, aquaporin 2 rather, and they do increase with vasopressin. And in fact, on a percentage basis, the percent increase in aquaporin 2 in the wild types and the knockouts is the same. But it does the animal absolutely no good in terms of concentrating its urine. So we came up with a different kinase to test, AMP kinase, or 5' activated protein kinase. And many of you are familiar with this because AMP kinase plays a role in cellular energy balance. So why would we look at it? Well, I read a very nice review paper by these authors here that quoted data that AMP kinase can stimulate NKCC2 independent of vasopressin. And furthermore, osmotic stress can stimulate AMP kinase. And I had one of those aha moments where I said, oh, a kinase that doesn't need vasopressin and can increase NKCC2. I wonder what it does to aquaporin 2 or UTA1 in the inner medulla. Went back to my lab group, asked this question, and no one had ever looked. At least no one had ever published anything to address this question. So we did. And because in the interest of time, I won't show you a lot of the data, but I want to show you some of the key findings. And again, in tribute to Dr. Berg, I picked out the perfused tubule data. So here's a study where Anna Wang perfused intermedullary collecting ducts from rats, measured in this case osmotic water permeability, and added metformin as an AMP kinase activator. And you can see within 30 minutes there's a very significant increase in osmotic water permeability. There is also an increase in urea permeability, again metformin in the orange, and she used a second AMP kinase activator, ACAR, and got the same result on urea permeability. Well, it's great to increase the transport. What about what a patient would care about? Will they make more concentrated urine? So Orhan Efi did this study in which he took rats and treated them with tolvaptan. Now, you know tolvaptan is used to treat polycystic kidney disease, and the main side effect is that the patients get nephrogenic DI. So we took advantage of that as a NDI model and did this study. You can see rats, they start with the urine osmolality between 1,500 and 2,000. Orhan then put them on tolvaptan for a few days to make sure the V2R was really inhibited. Urine osmolality drops to around 1,000. And then he either kept them only on tolvaptan in the blue, and urine osmolality doesn't change, or he added metformin while continuing the tolvaptan. And you can see on day 6, 1 day of adding metformin, there's a trend to an increase, but from day 7 out to 14 in this study, the urine osmolality is back at the basal level and it's sustained. We didn't see any drop off in the effect, and it was persistent for this 8 days of study. Well, you might say we had reached the point where we had our answer, but metformin, unfortunately, is not going to be the answer to treating patients with NDI. And the reason is that metformin is a weak and a non-selective AMP kinase activator. And the amount of metformin used in our animal study, and I didn't really tell you this in the previous slides, but in full transparency, is just well above the equivalent maximal allowable dose for human use. We didn't think we couldn't give as much metformin to a person as we were giving to the rats. And so the other is, of course, metformin is an oral hypoglycemic agent. So we needed something better. And this is where sometimes serendipity comes into science. I was introduced to a pair of medicinal chemists in Georgia who were working on AMP kinase activators for the treatment of diabetes mellitus, and they had some that just didn't work for them. And we spoke to them, and I said, you're studying the wrong kind of diabetes. It's not diabetes mellitus, you need to look at diabetes insipidus. So they sent them to us. Now, bear in mind, there was no CDA, no MTA, none of the whole alphabet soup of what you're supposed to do. They sent them to us, and we tested them, and they worked. And then Janet Klein went on and tested them in the animals. So here, again, similar to the previous study with metformin, we're now using this molecule NDI 5033. Here is percent of control. You can see this is the basal urine osmolality. We measured tolvaptan only, and then this yellow line is set to basically taking the tolvaptan only and making that the 100% mark, and then added NDI 5033. And on virtually every day for over 2 weeks, you can see urine osmolality is much higher in the NDI 5033 treated animals than in the tolvaptan only animals. Not sure what happened this day, but animal studies, you always get, Dr. Berg always used to say every study should have one outlier, and I guess that day was ours. So here's our hypothesis of how AMP kinase can substitute for vasopressin in nephrogenic DI. Here's a normal cell. Vasopressin binds to the V2R, activates adenylyl cyclase, stimulates cyclic AMP production, activates protein kinase A, which ultimately leads to the phosphorylation and apical membrane accumulation of both aquaporin 2 and UTA1. AMP kinase is present. We've done Western blots. It is there, but it doesn't do anything. It's repressed by pKa, and it just sits there. It's like that little spare tire in the trunk of your car that you hope you never have to actually put on your car. But when something goes wrong, you get you blow a tire, you can take it out, and that's kind of what happens here. In NDI, vasopressin can't, if the V2 receptor is mutant, it can't bind to it, it cannot activate adenylyl cyclase. Lithium does the same thing. You get no stimulation of pKa, the repression of AMP kinase is relieved, and with the stimulus of either metformin or NDI 5033, you get the apical membrane accumulation of UTA1 and aquaporin 2. So in summary, urea permeability is stimulated by vasopressin through increases in UTA1 phosphorylation and apical membrane accumulation. Hyperosmolality is a non-AVP pathway to increase urea transport, and AVP activators increase water and urea transport and hold promise as a potential therapy for patients with congenital NDI due to V2 receptor mutations. It will not work for aquaporin 2 mutations, but it will work for the 90% of congenital cases, or might work, who have V2R mutations. And so perhaps my final take-home point is shown here, which is that understanding basic physiology of urine concentration has led to a potential novel therapy that we hope we will be able to advance and get into the clinic, but it is through the performance of basic research, basic physiology, that one can elucidate pathways, elucidate mechanisms that then may suggest targets for therapies for diseases that at present we don't have any therapies to offer to our patients. So the final slide, I've tried to mention many of the people who did the work. These are the individuals who contributed to the data that I've shown you. Again, Janet Klein is in bold because she contributed to everything. Greatly appreciate the support of NIDDK over many years, not just these studies, but throughout my career, as well as support from the Coulter Translational Research Partnership and Georgia Research Alliance. And again, a sincere thank you to the ASN for this award. It's very meaningful to me because, as I said at the beginning, I was inspired by Mo Berg's Homer Smith Award talk. I was mentored by Mark Knepper, who won the Homer Smith Award. And so to follow in their footsteps is really very meaningful and very humbling to me. Thank you for your attention.